this deck, and then we'll just roll right into the second deck on methods of microbial control. So this will be super fun stuff. We're just talking about mutations, how they happen, and then we're going to roll into the last little segment on talking about methods of genetic transfer, mostly between prokaryotes. All right, so with mutations, we are talking about how changes in nucleotides would affect ultimately the proteins that are expressed from those genes. Even these really subtle, seemingly insignificant changes that might happen could have pretty significant effects. And this is because proteins can have very specific functions. This example has a bunch of different proteins on this slide, and they all have structural characteristics. If you've studied cell biology before or done a course in protein biology, you know, like, these features are what's responsible for conferring whatever functionality these proteins have. Like this rhodopsin, this would be in the rods in the back of your retinas. This is like a photosensitive molecule. These are alpha helices of amino acids that are wound together in such a way that they are able to respond or react to light that's coming into the eye and adjust according to like dim conditions. Telomere proteins, you see a couple alpha helices, but then you see these random coiled regions here. Leucine-rich repeat patterns, kind of an interesting molecule. You see this a lot with toll-like receptors, which you guys have learned about already. So it's important for them in their sensing function. This is a beta barrel. This is structured for nucleoside transport. So it's just a specialized protein molecule that allows for the transfer of nucleotides through these pores. Oftentimes in eukaryotes, when you're trying to transport nucleosides from the nucleus into the cytoplasm. Cadherin, another example. This is important for attachment and cell-cell attachment. And you kind of get the gist. All of these are specifically organized and coordinated in such a way to function properly. So if you mutate one amino acid in one of these proteins, which might be 400 or 500 amino acids long, you could destroy its functionality, especially if you're talking about enzymes and the active sites and enzymes. All right, first we have one example that we're going to drill down on, and that is starting off with a viral protein. This particular viral protein is the viral receptor on HIV. So, and this kind of illustrates this point, what happens with selective pressure. We're going to be talking about mutation rates. With viruses, mutation rates tend to be quite a bit higher than they are with bacteria, and certainly quite a bit higher than they are with eukaryotes. So HIV, for example, it's an RNA virus, which has super high mutation rates. And when these viruses encounter something that it has like deleterious effects, bad effects on it, it'll kill most of them, right? That's a negative selective pressure. But the mutation rates themselves don't change. It's just you get selection for those mutations that happen to confer survival through those adverse environments. So going back to this example here, in the lab, these researchers took HIV particles and they exposed them to this antiviral compound, whatever it is, tenofovir, abacavir, some like antiretroviral compound, at increasing concentrations. And then they waited for a period of time and then looked to see what kind of mutations had accumulated in the survivors, in this portion of that population of viruses that survived. So initially, this viral glycoreceptor was, this is wild type, meaning it's a normal molecule, hasn't been mutated yet. No change, but after a while, they started to see a single mutation, a V82A mutant. So there's a valine in place of an alanine in position 82 of that viral glycoprotein. And because of that one mutation from a valine to an alanine, which is a relatively conservative amino acid change, suddenly you get a phenotype. Phenotype being an actual pronounced change in how this viral receptor works. And that phenotype was resistance to that drug, 4 times more resistant, because you've made that amino acid switch from a valent to an alanine. More time passes, you have more selective pressure on these viruses to mutate the receptor. Now all of a sudden you've got 3 different mutations. You've got this valine mutation, you've got a methionine in place of an isoleucine, also fairly conservative. a phenylalanine instead of a leucine, and those 3 mutations now confer 11 times the resistance relative to the wild type protein. And then finally, we see one with 6 different mutations, all of this protein. They're indicated by these different colors, and we now have 30 times the resistance relative to wild type. So this is done under laboratory conditions, but this is exactly what happens in the real world too, when you put selective pressure on viruses and on bacteria to make these mutations and force it to adapt. Yes, sir?
Sorry, could you just repeat, or, let me clarify my understanding. E82 alpha means valine with alanine or NF?
You had it correct. Valine is instead of alanine at position 82 in however big that particular viral protein is. Yeah.
So you said this happens kind of in the wild. The people who have kind of been in the medical field for a long time, is this what they're talking about? They're talking about superbugs, like don't overprescribe, Z-Paks? Because this happens.
Yeah, it's very related to that. It's just one, biologically, again, like it's not necessarily that the virus and bacteria is deliberately changing in response. It just has mutation rates. And we'll go over that in a second, like what those mutation rates are. But over time, given enough selective pressure, those survivors, their populations expand because they are the ones that are able to overcome whatever adverse environmental condition, whether it be something natural, whether it be an antiviral, whether it be an antibiotic like a Z-Pak. Yeah.
Isn't that also how they got like different variants of the virus, like the ones that were more infectious but less deadly?
Absolutely, absolutely. So, not that I followed this very closely during COVID, but back when alpha was still circulating heavily, I can't remember the name of that first major mutation. It was like a D82I mutation or whatever, but it was in the spike protein. And oftentimes these mutations do occur in the viral glycoproteins. Those are the receptors. That's where a lot of these selective pressures are. And that lasted for a while. It became dominant in the population, as you guys all remember, right? And then there was more selective pressures, and these viruses changed. We got beta, we got Omicron. Omicron had a bunch of different mutations, not just in spike protein, but had mutations in the nucleic acid protein as well. So all those mutations had pronounced phenotypes that changed and altered the fitness of SARS-CoV-2 in this population. Okay, and then most of the time when we're talking about viruses and bacteria especially, these things are replicating and reproducing at very high numbers. With viruses, we're talking about millions and billions of replication events, and quite a few for bacteria as well. So when you're making mutations, these happen a lot. Most of them are going to be bad. Most of them are going to destroy protein functionality. And that's fine, because when you reproduce at high numbers like that, you can afford to have a lot of losses. We can't. That's why we have very high fidelity replication mechanisms. But these guys, absolutely. If you're going to kill off 99.999% of your population, that's okay. You can do that. Okay, so most of them are deleterious, most of them are bad for the pathogen. But what we see and what we notice, particularly when we're laser focused on pathogens, are the ones that confer advantages to those pathogens. Increased transmissibility, increased reproducibility, stuff like that, resistance to drugs. Okay, so now we have a couple of different categories of what causes these mutations in different pathogens. A lot of them are just called or considered spontaneous mutations. These would be usually oftentimes errors that are related to replication, like those polymerases that are coming along and accidentally, as you're going at very high speeds, high processivity rates, oops, you've inserted an adenine, you've paired it with another adenine. You've put a purine with a purine. You don't do that, right? So what that does is cause a distortion in the DNA molecule. If things are working right, you have proofreading capabilities and you can pluck that out with an exonuclease and fix it. But if they're not working right, then you're left with that incorrect base, and that gets copied on through progeny. It gets transmitted downwards vertically from generation to generation. So that's a big thing that can happen. You can fail to fix those things also, so your fail-safes fail sometimes. And I made a little bit of a correction here, but I was talking on the last slide with viral mutation rates with HIV specifically. And this is oftentimes how it's expressed. So this mutation rate gives us sort of like an indicator of how frequently we can expect mutations to occur in a particular organism or virus. So with viruses, for example, they have higher mutation rates, particularly RNA viruses, because RNAs has lower fidelity polymerases. With an RNA virus, a very high mutation rate would be on the order of 100,000 mutations per base per replication event. So that's more common. An organism like us, that has super, like a eukaryotic human, with our high fidelity replication machinery, our mutation rates are much, much lower, as you'd expect, right? Somewhere on the order of maybe once every 100 million mutations per base pair, per replication, per cell division, okay? So in this calculation, we kind of consider the size of the genome for that organism. Humans have 3.2 gigabases, 3.2 billion base pairs in our genome. So you can kind of calculate our mutation rates, what you would expect to have, the number of mutations per cell division. But for a virus, let's say SARS-CoV-2, that genome is much, much smaller. So it has about 29,000 nucleotides in that RNA genome. And you can do the math. Your mutation rate is pretty high, like 1 in a million, and you have a genome that's on the order of 10,000, you would expect there to be, however many mutated base pairs per replication event. And also when you have a single cell that's being infected by a virus and producing lots and lots of copies, those mutation rates become exponentially higher. With HIV, again, going back to that example, when you have a human being affected, not in vitro, not in the lab, but an actual human infected with HIV, that virus will produce like 10 billion new virus particles per day when it's during an active infection. So all the while, this RNA virus has those mutation rates to deal with. Because of that, the number of types of viral mutations in one person with HIV gets to be ginormous. You have a lot of different types of HIV in one person. Okay, so spontaneous mutation rates. This is sort of like the clock that is set for different organisms. You can kind of rely on this happening at a fairly constant rate. You can extrapolate how many mutations might occur, what might be reasonable. And over time, yes, this is a driver of macroevolution. So over time, we accumulate mutations and we change, always. Yes, sir?
And does the replication errors, the virus using host material to build itself, does that add to the replication errors because we're not able to recognize information?
It depends whether it's an RNA virus or a DNA virus. If it's a DNA virus and it's in the nucleus, replicating its genome with our DNA polymerase, it's going to benefit from the high fidelity polymerase that it uses from us. But RNA viruses have to use RNA polymerases. They have to bring their own and code their own. And that's one of the reasons why RNA viruses have such higher mutation rates than DNA viruses, because they're just relying on those crappy RNA viral polymerases. Yeah. Okay, so for chemical mutagens, we're talking about alkylating agents, talking about agents that can interpolate into DNAs, like in between the bases in DNA, like rubidium bromide, acridine orange, things like that. All of those agents will distort and deform DNA molecules and cause it to mutate. We also have radiation as a major cause of mutations. It can be ionizing radiation like X-rays and gamma rays, these high energy forms of radiation that are so powerful they can induce double-stranded breaks in DNA. And then non-ionizing radiation, just exposure to UV light. Just simple exposure to UV light can induce things like thymidine dimers, so inappropriate binding of thymidines together. So that'll mess up DNA as well. So all of these things are responsible for causing these mutations. So let's look at a few examples of how this occurs or what effect this might have when we're actually talking about getting ready to go from a gene to transcript to protein that's translated. if you just have an error that results in a single nucleotide substitution, one base substitution, a couple different things can happen depending on where that mutation occurs. We've already studied codons a little bit, so remember that codon chart and the fact that those first and second positions of those codons are less tolerant to changes than that third wobble position is. So, for example, a silent mutation would be one where there is no change to the amino acid resulting from that change. No effect. An example here is if you have a codon CAA, which would encode for a glutamine, and in that third position, you have a mutation that instead of an adenosine, you get a G in place of that. So if you look at the codon part, if you go codon chart and you go from a CAA to a CAG, they both encode for glutamine. That's a no harm, no foul situation. No effect. Same amino acid. Let's look at another example where you're mutating the second nucleotide in a codon. Instead of CAA, you have a CCA. Now, if you follow that in your codon chart, you're going to get an amino acid change, going from a glutamine to a proline. This is where you have to have some sort of knowledge about what amino acid structures do, what kind of properties they confer, if you can predict whether or not that's going to have a major effect or a minor effect. But glutamine and proline are pretty different amino acids. This is a long linear amino acid. This is cyclic, has a ring structure to it, tends to be hydrophobic, alpha helical breaking amino acid. So more than likely, this would not be a well-tolerated amino acid change. Okay, and that was from just substituting that second position on the codon. Last example would be where you have a change, a single nucleotide change, which results in what we call a nonsense mutation. This is when you have your codon change to something that would result in a stop codon. So the first position, you have a C. If you change that to a U, you get a stop codon. What do you think that does to your protein? It's probably going to destroy its functionality. Yeah, and it depends where that stop codon is introduced. If it's introduced near the end of that protein, like near the carboxy terminus, maybe it can be tolerated. But if it happens earlier on, the carboxy terminus, probably not. Okay. So, looking at this cartoon here, just to make sure we're all tracking and how this is read. This would be kind of representing what happens during transcription, right? You have now your DNA, single strand, and an RNA polymerase is sitting there synthesizing mRNA, so it's bound to that. In wild type, which again, wild type just means normal. In wild type, you have this perfect matching complementary nucleotides from mRNA with that DNA sequence, which would be a gene that's being copied. And then you have a corresponding code of amino acids that are put together to form some functional protein. When you have a single nucleotide substitution, let's say instead of a cytosine, you now have thymidine, like that polymerase screwed up and put in the wrong nucleotide. What happens in this example is you now have an mRNA transcript that's screwed up. It has an AAA instead of a GAA. And instead of a glutamate, now we're going to have a lysine. So what effect will that have? We don't know. It's just one amino acid that has been changed. But which one of these types or categories would that represent? Missense mutation, exactly. Okay, so those are, yes?
We assign different names to when there's like these point shift mutations on DNA versus mRNA. Like, can each of them receive like their own deletion and insertion?
Yeah, you can make.
So you could do it in DNA, the replication, or you could do it while the mRNA is being.
Exactly. So in this example, you're seeing mistakes that are happening in DNA replication, right? But which do you think has higher error rates? DNA replication or
mRNA.
transcription? Exactly. 10 to 100 times higher replication rates. So more often than not, if a mistake's going to be made, it's going to be made transcribing from a gene. Yeah, exactly. And that again is just by virtue of the fact that an RNA polymerase doesn't have as high fidelity as a DNA polymerase. But with the word shift, that's not what is happening here. I just want to point that out too. The shift is what's happening here on this slide. So these are point mutations, base substitutions. And this slide is talking about where you actually have an insertion or a deletion of nucleotides. Why that matters oftentimes even more, is that now you've screwed up the entire reading frame that we talked about. Those codons are read by ribosomes in frame in order to produce a logical, sensible protein message. If you screw that up, you're going to foobar whatever protein you're supposed to be getting out of that. So let's look at that a little bit. When you have insertions or deletions of bases that shift the reading frame in multiples of 3, again, because that's, if it doesn't occur in multiples of 3, you're going to have a frame shift mutation. If it does occur in multiples of 3, you might miss out on an amino acid or two, but it's not going to be the entire message screwed up, as if it would be if you just shifted 1 or 2 nucleotides away. All right, so in this example, we have our wild type DNA, mRNA, and polypeptide schema put together. And here in the middle, we're looking at what happens if you were to insert a cytosine. So you have an additional cytosine inserted into your DNA. The DNA polymerase is screwed up and added this in there. So in that case, instead of glutamate, now you're going to code for GGA on your codon and get a glycine. But that's not the end of the story, right? Because you've shifted this entire reading frame. So now everything downstream of that is also screwed up. Does that make sense? Okay, good. So that's an insertion with a deletion. Similarly, in this cartoon, you're showing one of these adenines being plucked out accidentally. Your DNA polymerase forgot or didn't add adenosine. So now you have a threonine instead of tyrosine, and this is showing this happening a little bit upstream of that. So everything after that phenylalanine is going to be completely screwed up. Yeah.
And what do you mean when it says cannot occur in multiples of 3? Is that like 3 adenines get taken off at the same time, or 6 of them get taken off at the same time?
Yeah, so when we're talking about additions, insertions or deletions, like that could be a single nucleotide insertion or deletion. It could be 2, it could be 20, it could be 30. But what I mean is if it occurs in a multiple of 3, like 30, then it won't be a frame shift mutation. Yeah, it's still an insertion deletion, but it's not a frame shift mutation. Frame shift mutation is when when it's the number of insertions or deletions is not a multiple of 3, because then the codon message is preserved. Yes.
2 codons that we're going to make offers and then one another one. That's a good mutation.
Could you say that just a little louder?
Like
Sorry.
if there was, I guess I'm like looking at this like if there's UAC for tyrosine, but AC and the G is not glutamate or deleted, wouldn't this cause a friendship?
So you're looking at the codon here for tyrosine?
Like if 2 of those and like one of the next one, like that would still be a friendship.
Yeah, so if you're deleted like the C and the G? Yeah.
Like if there was 3 total, so we got
Yeah, any multiple of 3. If you were to move C, G, A, you'd probably alter this tyrosine, make it something else. But as long as it's a multiple of 3, you would have an A coming in here, and then that GAG would be preserved. Yeah, I'm not entirely sure I was tracking on which ones you were pointing to, but yeah. Okay, in terms of one other point there, these usually result in nonsense mutations, and also, in addition to screwing up the reading frame of that transcript, you can also get stop codons inserted as well. Okay, so that when you have a truncated protein, it basically is probably going to lose functionality altogether. Okay. Any other questions about mutations, frame shift mutations versus single nucleotide substitutions? Okay.
One quick thing. Are we gonna have the codon chart on the exam if there's any questions regarding mutations?
Oh yes, I meant to tease Levi about that yesterday. You do have to memorize that entire codon chart. I'm kidding, guys. Sorry, it's not a funny joke. Yeah. No. Sorry, terrible sense of humor. If I have a question that asks you to like extrapolate something from one of these different types of scenarios, you'll have a codon chart. And I'll have some practice questions during review for you on how we might use some of those questions. We might handle them. Any other questions? Okay, just a few more slides talking about mechanisms of genetic transfer. This is mostly prokaryotic centric, but there's some stuff about viruses as well. We're talking about these different mechanisms. First and foremost, we need to define plasmids, which we really haven't talked about yet. We've talked about the chromosomes of bacteria. You guys remember some of the major characteristics of a bacterial chromosome? Yeah, it's circular, nonlinear. We only have one copy of genes, meaning it's a haploid genome.
Non-circular, right? Just working around.
It's circular. It is circular, yeah. So, like this cartoon, it shows it as a messy little nucleoid molecule, but it's still circular. It does not have ends to it. So, yes, we have our bacterial chromosome. On average, they're usually between, let's say, 2 to 4 megabase pairs. Again, that's about, if you contrast that to humans, we have like 3.2 billion base pairs. So you have your bacterial chromosomes, and then you have these other genetic elements that are also considered part of the genome. They also have genes that encode for proteins that can be super important, and these things are called plasmids. They're usually anywhere between like 100 to maybe 10 times smaller than the bacterial chromosome. So they're on the order of 300,000 down to maybe 50,000 base pairs of plasmids. They're also circular. They are defined as being able to self-replicate, meaning they do have their own little origin of replication. And when that parent cell is ready to divide, those plasmids will divide as well. They can be present in different variable copy numbers, meaning that sometimes those plasmids will copy themselves and make 5 copies. Sometimes they'll make 100 copies of themselves. But those copies in a perfect world should be split relatively even between those daughter cells. They don't always get split evenly, and sometimes they dilute out altogether if they are too burdensome for that bacterial cell to carry. We'll talk about that in a little bit as well. In terms of the types of genes that are encoded on plasmids, oftentimes this is where bacteria will pack a lot of their virulence factors. What does virulence factor mean? What is virulence in the first place? Yeah, it's something that's, at least in the perspective of human infectious disease and clinical microbiology, virulence factors are factors encoded by that bacteria that would be damaging to us. Things like toxins, for example, bacterial toxins. So a lot of bacterial toxins are encoded on those plasmids. You also get things like genes for antibiotic resistance. Those, neither virulence factors nor antibiotic resistant genes are essential for this bacteria. They're the extra bells and whistles that it carries with in order to cause infections and live under adverse conditions like heavy antibiotics. So, yeah, that's a defining characteristic. And this note about genetic engineering, for those of us in the sciences, we oftentimes manipulate plasmids and do a lot of our research using plasmids. So we can code for specific genes of interest, put it into bacterial cells, study the effects, have the bacteria mass produce those proteins for us, whatever we want. But we oftentimes use plasmids for research purposes as well. Okay, so now that we know what plasmids are, we can talk about different types of genetic transfer between bacteria. So these 3 different types of genetic transfer are common to bacteria. These are all bacterial methods of gene transfer. Oftentimes the word recombination gets thrown around interchangeably with genetic transfer, but recombination is a little bit different in terms of its definition. Recombination means that you've actually managed to integrate these genetic elements into some donor cell genome. Okay. So we'll kind of define what that means in a couple of examples. But the end result is that now you have a bacterial cell that's received some genetic element from a donor cell. You have a recipient that's received information from a donor cell. And you have a bacteria that now has a new type of genome, something that's altered its genomic composition. first method we're going to talk about is conjugation. This cartoon is from the Murray textbook, and it shows two different types of conjugation happening between two bacterial cells. These bacterial cells, the donor and the recipient, the donor being the one in orange and the recipient being the one in blue, they don't have to be the same species in order to make this happen. And not all bacteria can do this kind of conjugation. But the point is, or at least the gist of it is, they can swap out plasmids, so the donor can make, donate copies of its plasmids to the recipient. And they do that by forming this structure that is fimbria, but it's this long, specialized type of fimbria that we sort of touched on earlier, that's called a sex pilus, a tube that's connecting these two bacterial cells. Not very intimate. This is an actual picture of a donor and a recipient, but it's a very, very long pilus reaching out from donor to recipient. And what is going on in that tube is that you are transmitting either a plasmid, like you can see on the left, or parts or pieces of the actual bacterial chromosome are going across that sex pilus. Eventually, that piece of acquired genetic material, if it integrates into the recipient's genome, that would be recombination. Okay. Yeah, it is kind of like that. I don't know how much communication or consent is going on between the cells. It's similar. One thing though, like this has nothing to do with bacterial reproduction. So these things are going to be dividing whenever they want, and then this can happen at any time. It's more like, unlike sexual reproduction with humans, for example, where you're doing that in order to increase genetic diversity in your offspring. With these guys, it's a little bit more casual. It's like just an exchange of information. Okay, has nothing to do. I mean, if it's integrated, yes, the offspring of this recipient will receive that genetic material as well, but it doesn't have anything directly to do with bacterial reproduction.
If one of them has like DNA, the other one has RNA, can the conjugation still happen?
RNA. So the genomes are always going to be DNA genomes, chromosomal DNA and plasmid DNA. And the RNA is just doing the same functions it always does. It's either messenger, ribosomal, or transfer RNA. Yeah.
Is there any biological advantage for bacteria transferring like antibiotic resistance of its plasmid to a different species? Wouldn't that just increase its competition?
That's a really good big picture question. And actually, like, we'll talk about that with Enterococcus. Enterococcus is one of those really chatty, it's a genus, but like Enterococcus faecalis or Enterococcus faecium, for example. Chatty bacteria that have a lot of antibiotic resistance genes, particularly to antibiotics like vancomycin, VRE, vancomycin-resistant enterococci. They like to share their vancomycin resistance genes via conjugation. And they do so with competing species, which is weird, but that's what they do. They'll share that information with species like Staphylococcus aureus. Species that up to that point weren't resistant to vancomycin will receive those resistance genes from Enterococcus. So from a community population perspective, I don't know why the Enterococcus would do that, but it is done. And it's done on an interspecies basis and interspecies as well, from Enterococcus to Enterococcus.
What would be the opposite of recombination? Like, is there a occurrence where the DNA does not integrate?
Yeah, so this is a little bit of a gray area. So you have like the main bacterial chromosome. You can have integration of entire plasmids into that chromosomal genome, or those plasmids can stay unintegrated but still actively being transcribed and producing products that do stuff, right?
And that's still recombination?
I don't know. I'd have to look up and see exactly whether that still counts. It's episomal, which means it's outside of the chromosome, but still counts in terms of changing that bacterial cell. I don't think it actually counts unless you have integration into that chromosome, though. Yeah. So if you pick up a plasmid, this is not really necessarily a permanent thing. You can lose plasmids over time as well. If you think about plasmids that, I don't know, carry genes for toxin production or other virulence factors, I mentioned this earlier, at least alluded to it, those do come at an energetic cost to the bacteria. So if they don't absolutely need to carry this big, heavy antibiotic resistance gene to vancomycin, if there's no selective pressure, it's better for the bacteria to just dilute out those plasmids over time, which kind of is a little bit of a preface to what I was going to talk about next lecture when we talk about antibiotics a little bit. And the fact that that does give us hope. If we lay off antibiotics over time, that reduces selective pressure on bacteria to carry antibiotic resistance genes because they do lose those plasmids. Not all of them are encoded on plasmids though. Some of them are chromosomally encoded, so you're not going to get rid of them that easily. Okay, second method of genetic transfer between bacteria is called transformation. What this cartoon is showing here is that you have a normal, healthy bacterial cell with its own genetic information. That cell dies, and when it dies, its chromosomal material and its plasmid material fragments and gets taken up naturally by a completely different bacterial cell. So again, this doesn't have to be the same species. It just is taken up through the cell wall, through the cell membrane, and is integrated into that recipient cell. This happens quite a bit in nature, and this is another thing we do a lot in the lab. Has anybody ever worked in like a research lab before? Done some transformations before? Okay, cool. So yeah, we do this a lot in lab where instead of just naturally allowing these bacteria to take up information from a donor, because this is an arduous process for the recipient, we will force these cells through electrocuting them or through heating them or just whatever forms of stress we can put on these bacterial cells, and it forces them to take up more DNA than they normally would. So those are called competent cells. We can make those cells more competent, which means we increase the amount of DNA that they can take up. Ultra-competent cell lines. But in nature, you also have competent cells. And not all bacteria are able to take up this naked DNA that's floating around as well. All right. Let's just get through transduction. I'll put you guys on a break after that. Okay, so this we haven't really talked about before, but there is another very significant major category of genetic transfer, and it is facilitated by viruses that specifically target bacteria. These viruses are called bacteriophages, or just phages for short. What do you think the word bacteriophage means?
Eater.
Yeah, eaters of bacteria. It's not actually eating them at all, though. It's not eating them at all. It's just injecting genetic material into those bacterial cells and then making copies of itself just like it would with a eukaryotic cell. So we'll talk about that in a little bit more detail. But first we have to talk about, oh, and I did just put this on here. It's a nice image of some bacteriophages here before we talk about how that happens. So these little red things are bacteriophages, just viruses that infect specifically bacteria. They have these tail fibers on the end of them that are specific for different types of bacteria. They come and clamp down, and then they force this injection of genetic material into that bacterial cell. This is all viral material. All right, so with that image in our head, this is how phages can manage to increase genetic diversity with bacteria. So you have your phage that is injecting its own genetic material into a susceptible bacterial host cell. You have it entered into that bacteria. If it enters the lysogenic cycle, which we'll talk about first, that's the part on the right here, that viral DNA will integrate into the bacterial chromosome. So we have actual recombination occurring here. That bacterial chromosome is now altered to carry viral DNA. That's called prophage once it's integrated into the genome. And when that bacterial cell divides, it's going to also make copies of that viral DNA as well. If there are genes on that viral DNA, those will get expressed, but they're probably going to be expressed at low levels. And this lysogenic cycle can continue on stably for some period of time. After a while, or depending on whether or not it gets like a specific environmental cue or some stimulus, it can enter what we call the lytic cycle, where that prophage DNA excises or pops out of the bacterial chromosome, and it starts really actively transcribing viral components. So it's replicating its genome, it's making viral proteins, and then eventually when you get a critical mass of all these viral components, that bacterial cell, the cell wall will weaken and that whole thing will burst or lyse. That's the lytic cycle. So that is going to release a bunch of little progeny bacteriophages that can go on and reinfect other bacterial cells. Where this is important for transferring bacterial information is kind of right here. I want you guys to look at this at the bottom left. Sometimes during the lytic phase, when those viruses are assembling their genomes and their viral components, sometimes they will accidentally take up pieces of the bacterial chromosome. Some of those pieces may have entire intact genes that encode for things like toxins. Okay, so that's kind of what's happening here. An example of that is the cholera toxin phage that encodes the entire toxin gene. When you have a phage that infects a cell that's carrying that gene, for example, it is suddenly going to be able to express cholera toxin. Okay? So these phages have sloppily, accidentally packaged parts of the bacterial genome, and it contains genes. The new bacteria that get infected with bacteriophages will also be expressing those bacterial genes as well as the viral genes. Okay, and then the very last type of genetic transfer we're going to talk about, it has nothing to do with bacteria. This is a viral mechanism, and this last method is called viral reassortment. This happens between viruses that have segmented genomes, so pieces of their DNA on different segments, and it has to occur in the same cell. So what happens is you have virus A and virus B. They're infecting the same susceptible eukaryotic cell, and when they're infecting that cell, those segments will shuffle together, and the result is you have a completely new type of virus. This happens a lot with influenza, and this is what is one of the primary drivers of those pandemic strains of influenza, where you have completely new combinations of influenza viral segments, genomic segments, that have been reassorted together into one novel virus. We'll talk about that more in block 4, but that's like a very, very important characteristic of some of these segmented viruses like influenza virus and like rotavirus. Fortunately, most viruses are non-segmented. They don't have segmented genomes, so they're not capable of doing that. But influenza is an exception. Okay. Yes, sir.
So, for the sake for this, is this something like how Spanish flu is built? Like you had one subflu, you had another subflu. Spanish flu. That's why it's
Yeah, exactly. Yeah, so like we had at least 5 major pandemics of influenza in the 20th century, right? With the 1918 Spanish flu, you had a reassortment event happening in one infected cell and a new influenza virus being produced as a result. That new influenza virus was something that the population didn't have immunity to because it hadn't been exposed to. This is before we vaccinated people against influenza anyway. And that particular type of influenza virus came out of that. It was just a very virulent virus, and it was very transmissible among the human population as well. Okay, I've got 10:46. Let's take a 10-minute break, and we'll start back up at 10:56. Describing common antivirals or also overused antivirals, then selective pressure. Yeah, I mean, you cannot grow viruses that are out there. Yeah. Not all trial and error.
So I was just looking over these. Does also guide when it transfers all of that genetic material, so it leaves some genetic material for itself?
Yeah, so that's the point. Plasmids like they have a specific
Is that a two-way street then? So does the receptor itself also transfer?
as far as I know, it's a recipient as well. Some of them not all.
So the conjugation is being
Yeah, that's true.
Yeah, but dog.
I feel bad also, but like I feel like I'm sometimes I do something and like no 100%, yeah. And then I like during the weekends I take a walk and have my phone I'm trying to figure out what to get some energy out. I know he's pretty busy too, so yeah. So like I can't go for runs. It's not like they're like rapid. It's actually like a 3 tours of the hour. We have our good taxi. Oh yeah. I would get another one like that. I run it right side, my I mean, that way, like, that's fine, you know, like, once I can, yeah, the black the other one I got there, so initially we were thinking one day, there's an airport so we changed it to like the next day. They're like, so if we change the flights, drop them off, got on the plane, all over, I'm like, one of those was called that, so I was looking at my phone, I had to get it. I'm like, and I like texted what's going on with the dogs and stuff like that. They sent me in when I was the collar probably fell to the ground and I'm bored. Where did they respond? So we take off, we flight. I'm like, I talked to the stewardess over there. And I'm like, hey, check the plug after the job thing. So I got to Austin, sorry, Dallas. You buy a lot of stuff with that one? Yeah, they should hang out more. Yeah, but it wasn't like stuff like that. Well, I did pull my couch. Not causing, but I thought about that. I should. Go have a fun. I go back. I know, that's the story.
Stop the squish. Those are squishies. Huh? Those are squishies? Yeah. You call me a snowflake? I gave one to Taylor too. I was like, you guys are snowflakes. I'd eat my yogurt.
Yeah, I heard it was dirty. Yeah, I think it was a good place. I went there once or twice. First time we go there, we just like an hour. Yeah. And I promised myself I would go twice. And then his mom has like
I gave it to Taylor. She's like, why am I the snowflake? She's like, no, Lucas is a person.
You could study. Studies before. That's why I asked
do your job. Unless you speak Spanish,
Yeah.
Germans are rude too.
I usually sleep on the
Everyone was nice.
Oh, Germans are blunt. French are awful.
Actually, do the same thing.
I was like,
And then it's my
she
parents.
actually like, normally like she's through like 4 alarms.
Because they want you to
Just far past now. She finally woke up.
And if you don't, they're like,
I was like, checking down. She
I don't know if I was like, what? That lost.
she's like,
But I'm sure because we they're a lot
I
nicer.
just figured one of them. Wow, you woke up spicy.
I also heard that,
She was like,
I think they do that here in
I like that. But if like I was studying, Sailor's easy.
Did you go to Spain?
She was all about
I guess I better download the next one.
What's in my
Huh?
like
I guess I better download the next one.
people go to Ibiza to party for my own fans for like
What is this one? Microbial growth?
Watch movie. Study directions. I'll tell you if you turn it off. I was like, turn it off. Turn it off.
Okay, guys, let's pick back up on the second deck, starting control of microbial growth. Main point here, we just want to get familiarized with kind of disparate topics, but the common theme is these are things that kill, that we have in our arsenal to kill microbes and toxins and viruses. Yes. So we'll talk about some terms, just the usual format, talk about things that would affect the efficacy of these different disinfectants and sterilants and antibiotics, talk about which specific microbial processes are affected or targeted by them. And then the last part of the deck, which we definitely will not get to today, is all about just like a primer for antibiotics, how they work and what they're targeting. All right, so today we'll get through all the biosafety stuff on this outline, and I think we'll make most of the way through the chemical disinfectants, but tomorrow for sure. Well, we won't start in physical control today. Okay, so just a couple slides to kick us off on biosafety and what that means, biological safety programs. This is one of those oftentimes taken for granted features that if it's working well, no one really knows it or appreciates it. Oh, there's an enthusiastic head nod. Yes. Yeah. And when it's not working well, then we get a lot of negative attention on these things. Anybody work at USAMRIID before? A couple people? Okay. When did you guys go to USAMRIID?
We just came there.
Oh, okay. Okay. And when did you start that tour?
23 from earlier.
maybe 21.
21? Okay. So I got there in 2018, right after I finished my PhD. I did my postdoc at USAMRIID. And that was, if you know anything about the year, like 2018 to 2019, those were dark times at USAMRIID, right? USAMRIID is the U.S. Army Medical Research Institute for Infectious Diseases. It's kind of like the mecca for 71 alphas, and I'd wanted to work there my entire life. When I got there for my postdoc, USAMRIID was basically in a downward spiral of funding and biosafety infractions and basically being shut down. So all of that really cool, exciting research that I wanted to do was basically shut down. And it just was like a negative feedback loop because of our shutdown research. We couldn't attract new partners, we couldn't get external funding, and it was very, very hard for USAMRIID to dig itself out of that hole. But yes, sir.
Is that kind of stuff monitored by the CDC?
It is, yeah. So USAMRIID is a BSL-4, which we'll talk about in a second. It's the DoD's only biosafety level 4 lab. You can't just run in a vacuum. It doesn't matter how cool you are. You still get inspected by an outside regulatory agency. The only regulatory agency in the United States that can inspect USAMRIID is the CDC. So they came in and they started inspecting us, and they found that we weren't adhering to our own safety protocols, our own biosafety protocols, and shut us down. That's how later? Yeah. 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 So biosafety, all that to say, biosafety is kind of a topic that's near and dear to my heart. I think most of us who worked in a lab before have stories about, the time we were working with insert your pathogen of interest and you almost got killed because you didn't realize it was not a vaccine strain or something like that. I mean, lots of crazy stuff happens. So we try to put in as many layers of different controls as we possibly can, and yet we still have accidents happening all the time. Some minor, most are minor, and some of them are major, like some kind of legendary, epic biosafety accidents. Anybody heard of the Sverdlovsk accident that happened where anthrax spores were released in Russia? Yeah, it was a lab accident in a research lab, and a bunch of anthrax spores were aerosolized, released into the environment, and they wafted downwind into communities, and I don't know, like 68 people got anthrax because of that lab accident. So it does happen. And each time we have some sort of incident or example of a failure, catastrophic failure, or even minor failure, we're supposed to look back and reflect on what went wrong and how to fix it in the future. Because of that, we have kind of a hierarchy of controls. It's good to understand the CDC triangle and how it's meant to function. But when we think about biosafety, a lot of us only just think about wearing gloves and washing hands and maybe wearing a face mask. But that's the tip of it. That's considered the very least effective way. You need a whole bunch of more intrinsic controls in order to be safe. Administrative controls, this is one of the weaknesses at USAMRIID at the time. They've corrected it, done a lot better. Bad SOPs, and people weren't following SOPs. Those types of administrative controls, things like that. Engineering controls would be building controls, making sure that the airflow in the room is adequate for the work that's being done. If you're working with select agents and pathogens, negative pressure rooms to make sure those pathogens don't escape. Circulating air at a certain volume, like 6 exchanges of room per hour, whatever the rates are. All of those are engineering controls. Wherever possible, if you're doing work with, especially if you're doing research on infectious agents, if you can use a substitute, use a substitute. Don't use the real deal unless you have to. Unfortunately, for publication purposes, you oftentimes have to go and use authentic viruses and bacteria to publish in high-impact journals, but you do the majority of your brunt work with surrogate systems if possible. When I was getting my doctorate degree, I was doing work on Ebola and Lassa virus, but I was doing this at University of Virginia, which is not a BSL-4 at all. I definitely wasn't working with those viruses there. I was working with pseudoviruses that had non-virulent scaffold viruses, but they were vectoring the glycoproteins of my virus of interest. So all of my experiments were done, all of my data was collected using these surrogate systems, not the real deal. And then when I got to USAMRIID, I started using authentic viruses. And then finally, elimination. So if you can remove yourself from the risk altogether, that is preferable. Such as in this fine establishment, this luxurious building that we're sitting in right now, where is completely pristine and there's no infectious agents here. Just kidding. In terms of key elements, I think I hit upon most of these, but in terms of risk assessment, for the people that are going to be working not just in research conditions or in clinical labs, but also clinicians themselves, like you guys, your ability to understand the risks rests on like your primary education, your knowledge of the nature of these different microbes. And that allows you to at least have an idea of what is potentially a hazard or not. Is something going to be a threat or isn't it? So that's part of the reason why we go to school and learn about stuff that seems like it would be otherwise pretty esoteric, so we have an understanding of the risk. Why is it important? Most obvious reason that we practice infection control and microbial control is to just prevent disease. Definitely we want to protect the community, we want to protect ourselves. If we're working in a lab, we want to avoid lab-acquired illnesses. Food safety industry is important. We want to minimize foodborne illnesses. We'll probably always have them. But if we have good regulatory systems, we can at least reduce them. And then particularly for you guys, HAIs, this is an acronym we'll use a lot in this course, hospital-acquired infections or hospital-associated infections. Just things that you guys might be exposed to, or your patients, especially if they're inpatient populations, would be exposed to. So you're going into a hospital seeking treatment for one thing, and while there, because there are a lot of sick people there, you're picking up something at that hospital. We also want to make sure we protect public health, food and water safety, hygiene in public spaces. Again, using this building as an example of a public space that's immaculate and clean. You just have minimal, great hygiene here and minimal risk of transmission of infectious diseases in this environment. Outbreak control, also important. CDC has a big role in this to play. For maintaining sterile environments, medical procedures, there's an old skit that has a surgeon that leaves a rag in the abdomen of a person they've been doing surgery on. It's kind of funny, but obviously we've evolved our ways of doing surgery in such a way that we are as obsessive about hygiene and aseptic conditions as we possibly can be, because that's when patients are most vulnerable to getting contaminants. And even things like injections. You don't just give somebody a shot, you disinfect that site with an antiseptic before you put a needle in someone. With pharmaceutical manufacturing, this is really important. We have GMP, good manufacturing practices, that are implemented. Compounding pharmaceutical companies, places that are making a lot of these drugs. There have been some very high-profile contaminants that have been introduced to products. There was one about 10 years ago where this compounding pharmaceutical company was making some sort of drug for immunosuppressed patients, and the contaminant was some sort of fungal mold, and it caused a bunch of cases of fungal meningitis, some pretty rare stuff, but a multi-state outbreak resulted from a compounding pharmaceutical company having contaminated fungi. I think it was aspergillus that made its way into that product. But yeah, like the most vulnerable patients were getting introduced to these fungal pathogens. And then lastly, research labs and clinical labs. GLP and GCP, good laboratory practices and good clinical practices, also very important. Making sure we do not introduce contaminants where we don't want them, we can't afford to have them. A lot of text in these early slides, so sorry about that. So the main point of biosafety measures is to just make sure we reduce infections and transmission. These 4 levels, BSL-1 through BSL-4, if you haven't heard of them before, they are mostly talking about labs, but they have associations to the risk groups of the pathogens themselves. So it's good for you guys to know and have a rough idea where these different pathogens fall in terms of the risk that they present to people working with them. The lowest level of BSL-1 involve pathogens, or non-pathogens rather, agents that are typically not associated with causing disease. E. coli, most of you have heard of before. There are definitely pathogenic strains of E. coli, like K1 is a very, O15787, a bunch of different pathogenic strains. But other strains like K12 are perfectly benign, non-pathogenic, they cannot cause disease. So we would work with them very safely in BSL-1 condition, BSL-1 lab. Even in those conditions, like we're talking a basic introductory college level course in microbiology, like a microbiology lab, even then they would encourage you to wear gloves, maybe wear eye protection if you're working with like a liquid broth or something like that. Of course, there would be a sink, and they would be stressing hand washing in BSL-1. Whatever bench surfaces you're working on would be non-porous, so you could clean them off easily, you could disinfect surfaces easily. But basic stuff. Take that up a notch, we're at BSL-2, moderate risk. Organisms like Staph aureus, HIV, Salmonella. Talking about HIV, that is a dangerous virus, especially it used to be, it still is, but it's pretty difficult to transmit, especially in a lab. You don't have a lot of lab-acquired HIV cases. We have very good treatments. Staph aureus can kill you. Salmonella probably can kill you, but these two, like we have very good treatments for them, so we don't have higher BSL protective measures requirements for pathogens like this. So for those practices, we'd maintain our BSL-1 practices and then include a couple extra safety measures. More signage, Sharps precautions. What are Sharps precautions? We don't want any accidental sticks, not for the person that is working with the instruments, not for the people that are cleaning up either. So if we can use those hard containers for anything that has a pointy end to it, needles, could be even plastic pipette tips. I mean, those things can pierce plastic bags. So you put them in a hard container, and it makes it safer for everybody. At this point, you would be using biosafety cabinets, which have directed airflow for your work. Self-closing doors if possible, so you don't have to handle them with your hands. And then eye wash stations would be available. With BSL-3, now we're talking about some more serious and exotic diseases. West Nile virus. It's actually becoming pretty common in the United States. I think we have it in all 50 states now. But it's still considered a pretty exotic virus. Mycobacterium tuberculosis, positive agent tuberculosis, can be a very, very serious infection. In the United States, we very aggressively try to control circulation of tuberculosis. B. anthracis is on this list. This is an uncommon pathogen. I hope you guys never see it in your careers. But we do have ways to treat it. It's not untreatable. And we do have a vaccine for anthrax as well. So in addition to BSL-2 practices, we would have decontamination of all waste. You don't just throw it in the garbage. You have decontaminate before you even put it in there. You don't just launder your lab clothing. There's special decontamination. If you can use disposable lab coats, that's a good idea instead of cloth lab coats. For respiratory protection, now you're probably talking about things like wearing PAPRs, which we'll talk about as well. Controlled air supply, clean air supply. And then a physical separation from access corridors, still self-closing doors, et cetera, et cetera. Okay. Finally, BSL-4's maximum containment. These are the worst pathogens. There's no level 5. So Ebola is on this list, dengue, Marburg, Lassa, Quinine, CCHF, Crimean Congo hemorrhagic fever, a lot of viruses where there are no good vaccines or treatments. Here, your personnel are going to be highly trained, very isolated units. You'll be working in class 3 biosafety cabinets or glove boxes if you're actually manipulating those agents. All the air handling systems will be specially tailored to handling those particular pathogens. background checks for personnel working with these rigorous health and occupational health standards for personnel working with them. You have to shower upon exiting, so pretty rigorous stuff if you're working with the BSL-4 select agents. And then there's another category that we all need to adhere to. It's called standard precautions. Yes, ma'am. Possibly, yeah, especially for emerging viruses, they're kind of in their own category of threat. Like the CDC has a special pathogens and emerging viruses branch to it, simply because when a virus is emerging, like SARS-CoV-2, when it came on the scene in 2019, 2020, we didn't have it characterized yet, so we certainly didn't have any medical countermeasures. We didn't have vaccines, we didn't have good specific antivirals. So that bumps it up into a higher category. And then we didn't have good data on mortality rates. So we didn't know if it was going to be killing 60% of the people infected or 2%, and that mattered according to how you stratify different pathogens. So yeah, there's like, with emerging pathogens, whatever they are, there's an initial period where you probably want to put it in a higher tier of threat than well-characterized, well-known viruses. And Ebola, for example, that has been a BSL-4 agent kind of as long as we've known about it. It's we don't have any really good specific treatments still for Ebola. It's a very virulent virus. It can kill people very quickly. We do have vaccines now, but they are not widely used. So it remains a BSL-4 agent, even after decades and decades. So with standard precautions, this simply means that in practice, to keep yourself safe, you do not know, and you cannot assume the health status of the people you're going to be around. And that includes the people in this room, much less your patients who probably do have something wrong with them or some sort of complaint. But everyone in this room looks well, everyone looks healthy. But if one of you were bleeding, I would assume that you're carrying a bloodborne pathogen. I would be careful about interacting with your blood, right? It's the assumption that you have a disease, even if you look well. So with that assumption, you adhere to these very basic infection control practices, and you use these practices all the time. You never deviate from them. Hand hygiene is the single most important measure to preventing the spread of infection. Cannot stress this enough. It's like the cornerstone of infection control policy wherever you go. Also super important, of course, PPE with gloves. And you don't just wear gloves, but you wear them properly. If you get a tear in a glove, you take it off and replace it. After you deglove, you wash your hands again. There's a proper way to wear all this stuff. And there are SOPs, like huge binders full of SOPs that give you careful instructions about all this stuff. We already talked about Sharps container, and then just maintaining a clean environment. If you're working in a lab, that's super important. You don't want it to be disorganized. You don't want it to be dirty. You keep that environment very clean. Okay, a couple terms to make sure we have a good understanding of. Chemical disinfectants versus sterilants. What is the difference between a disinfectant and a sterilant? Yeah, which one gets rid of everything? Sterilant, yes. So I did modify this a little bit, killing or destruction or elimination, it doesn't matter. The point is it's been removed as a risk factor. It's an all or nothing proposition with sterilization. You don't partially sterilize something, it's either sterile or it's not. With disinfections or disinfectants, the goal is not to remove or destroy everything, it's simply to reduce the level of microbial load to get it to a safer level. So this is designed to be a little bit more practical, pragmatic, economical perhaps. And in terms of semantics here, antiseptics are not quite the same as disinfectants. Antiseptics are disinfectants that can be safe for use on the skin. So if you're if you're using an antiseptic like povidone iodine, you're not eliminating all the bacteria on a person's skin, you're reducing it. It wouldn't be feasible or practical to remove all of it. Disinfectants are categorized as low, intermediate, or high level. A high level would be a disinfectant that is rated as like sporicidal or capable of removing like really difficult to kill or inactivate substances. Yes, sir. Yeah, exactly. So I have a chart somewhere in this deck. I hid it because it was kind of like an overwhelming amount of information. But on that chart, which you might be able to see, it had like low, intermediate, and high, and that's just some very common disinfectants to give you a sense of how they're rated. But on that note, you can start with a low-level disinfectant, and if you leave it on there for a long period of time, perhaps it gets bumped up to an intermediate. So there is a little bit of, or you can take a high-level disinfectant, use it improperly, and it gets bumped down to like a low-level disinfectant. But if you use it properly, there are assigned categories for those different disinfectants. Okay, so when we would use these chemicals would be in instances where we can't throw things away because that's too expensive. We can't heat them in an autoclave or something like that, heat and activate substances. And irradiation, for whatever reason, is not feasible. So a lot of different conditions and circumstances where disinfectants would be the way to go. On the left here, we see some sort of surgical instrument in this plastic pouch. Do you think that is disinfected or sterilized? Sterilized, exactly, yes. And on the right here, our old friend Lysol markets itself as being 99.99% effective in reducing bacteria and viruses. That's impressive, but when you're talking about millions of bacteria on a surface, you still have a lot of bacteria left at the end of a good disinfecting spray with Lysol. Okay. So it's a disinfectant, it's not a sterilant. Now we're going to talk a little bit about some of these factors that we've already started talking about with respect to chemical disinfectants, things that would affect how well they work, their efficacy. First, we have the concentration of the chemical. Intuitively, if you're using a chemical to disinfect something, you usually think that more of it and more concentrated amounts of it are better, right? And that's usually the case, but not always. So, for example, here, if you're trying to disinfect something with 95% ethanol solution, do you think that would be better than a 70% ethanol solution? You do? Okay, and that's intuitive, right? But it actually happens that 70% is more effective, usually. That's because the higher concentration of water in the more dilute formulation increases the penetrating power of that, and it also decreases the evaporation rate. If you have straight ethanol, that evaporates really quickly, it has a high volatility. When you water it down from 95% or whatever to 70%, much lower evaporation rate. So contact time increases, and you don't have to do a thing. So for that reason, if we kind of look at this, this chart right here and interpret that, first of all, we're starting with a flask of some bacteria on this cartoon, and we have a log scale on the y-axis, starting with 1 billion cells in that flask, and then on the x-axis, we're looking at time. So the reduction in the amount of viable cells in this flask over time. If you have 95% ethanol, the rate of disinfection is much, much slower. Again, logarithmic scale, so you're going from 1 billion to 10 million, 100,000, etc., all the way down. But much, much, much faster kinetics of disinfection with 70% ethanol. Okay. Similarly, we've mentioned contact time, but a lot of people, when they're using a disinfectant, they just kind of spray it on a surface and then wipe it away immediately. That might be okay for some things, maybe. It depends on the type of spill and the type of agent and the type of disinfectant. But let's say you have, I don't know, like somebody who's incredibly sick and they throw up all over the place, so you have like a huge amount of vomit sitting in front of you. I would highly recommend an extended contact time if you're going to be disinfecting something like that. Just because it's a lot of fluid and it's from a sick person, there could be viruses in that vomitus, like, so it does depend on the type of material that you're trying to disinfect, but contact time is always important. With all of these, the common theme is making sure that you read what the manufacturer's instructions are before you use that disinfectant. Temperature and pH. In general, as a general rule, most disinfectants work faster if they're at higher temperatures. But you have to be careful there too. You make sure that the disinfectant can handle higher temperatures. But intuitively, if you're trying to clean up a spill in a 4-degree refrigerated room, it is going to take a longer time because it's a cooler temperature in there. And that's what this graph is showing, much slower kinetics at 4 degrees than at room temperature around 25 degrees centigrade. With pH, that too is important. The chemical itself needs to be at the correct pH in order to work. If it's more acidic or more alkaline than it's designed to be, then that can affect its efficacy. And then the material that you're cleaning up as well is important. Using that example of vomit, that could be some pretty acidic stuff. So that might alter the pH of the disinfectant that you're using. Okay. Organic matter and biofilms, another important factor in determining the efficacy. Going back to surgical equipment, that has to be reused because it's really expensive. If you just put some scissors or a scalpel that's been like straight out of surgery into an autoclave, and it has gross organic material visibly on it while it goes into the autoclave, is that autoclave going to get rid of that organic material? No, it's not. It doesn't have sprayers inside the autoclave or anything like that. All it's going to do is kind of like cook that material onto that surgical instrument. So when it comes out, it's going to have caked on blood and whatever other bodily fluid is on that instrument. So before you put something in an autoclave that has tissue or blood on it, you have to remove that organic material completely before it goes into the autoclave. I have an example pretty soon in a couple slides of that too going wrong on an epic scale. Biofilms, we haven't talked about biofilms yet in this course. What are biofilms? Yeah, so a collection of bacteria on a layer that can be growing on substrates that you wouldn't expect, like implants, plastics, can support the formation of biofilms, but dense microbial communities, polymicrobial communities oftentimes, that reduce penetrating power of different disinfectants and heat as well. So that too, if you're autoclaving something with a heavy biofilm, it might be harder for steam to penetrate down into that equipment as well. Microbial load, I think this is the last factor that we're going to talk about today. But pretty intuitive. Again, going back to our flask with E. coli or some other broth culture, start off with 1 billion cells versus starting off with 100 cells. It's going to take a lot less time to kill bacteria if they're grown at a lower concentration. This is called less turbidity. So pretty intuitive, pretty straightforward. And I lied, there's one more category. The type of microbe that you're trying to disinfect. We already know, looking at this graph, the difference between spores and vegetative cells. What is that difference? Yeah, so we're talking about spore-forming bacteria here, like Bacillus anthracis or Clostridium. Those vegetative cells are actively replicating but more fragile. If they've happened to have sporulated and become spores, and you try to disinfect them, good luck. I mean, there are sporicidal disinfectants, don't get me wrong. They have to be rated that way. They're much, 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 much harder to disinfect, to the point where you might not be able to disinfect them with a given method. It might be impossible. You might have to switch to something else. Okay, so knowing what you're working with, and that is what this slide, I think this will be the last slide we talk. Yeah, we'll expand it today on this slide. But the nature of the organism is important for choosing the disinfection method. Prions are kind of on their own little level. They're very, very difficult to disinfect. They can withstand 121 degrees Celsius treatments, basically just immersed in very, very, very hot water for hours at high or low pHs and still retain infectivity. That's insane. Okay, so if you have material that's contaminated with prions, you have to have some serious decontamination procedures that you follow. Down that list, we have spore-forming bacteria. That's what this picture is showing. We've seen this before in the last block. Difficult to treat those spores. Then we've got Mycobacterium. They have that waxy cell wall. It makes it hard to disinfect those guys if you have Mycobacterium tuberculosis, other Mycobacterium species. And this is a little counterintuitive, but all of a sudden we see Staph and Pseudomonas, Staphylococcus and Pseudomonas. What is special about those two types of bacteria that make them so much harder to treat than other gram-positives and gram-negatives? Like if I said Staph aureus is a gram-positive and Pseudomonas is a gram-negative, anybody know something special about those bacteria that make them that resistant? Potentially.
Glycoprotein.
No, but good guess. Because others no. Yes, winner, chicken dinner. It is their ability to form biofilms. Yes, Staph aureus is a biofilm former potentially. Not all strains, but some of them can. And same with Pseudomonas. Pseudomonas is like king of biofilm formation. We'll talk about both of those later. But it just goes back to what I was saying about biofilms being intrinsically harder to disinfect. So after that, we've got cysts. We talked about protozoan cysts. Quite a bit more resistant than trophozoites. And we've got our gram-negatives, which are quite a bit more resistant than our gram-positives. They have more complex, robust cell wall than gram-positives. Fungi, fungal spores. And then down at the more fragile end, where it's easier to kill, we've got our viruses. Between capsid and envelope viruses, which do you think is going to be easier to kill? Enveloped? What? Yes. So capsid is what kind of material, what kind of macromolecule?
Polysaccharide. Oh, the boot.
It's a viral protein. Okay. Welcome to my world. Yeah, so capsid viruses have a protein shell on the exterior that does make them quite a bit hardier than envelope viruses, which are just about the most fragile pathogens you can imagine. Very easy to kill. Okie doke. Any questions for me about disinfectants or sterilants and biosafety levels?
What do you recommend for disinfecting agents within your own household?
I mean, Lysol.
She was home in a biosuit.
Yeah, I lick my countertops.
Oh, no, you use this stuff.
Yeah. But yeah, no, I mean, just, yeah, simple.
I relate this to like, if we're gonna be PAs and we're like in an austere environment, how do we disinfect? I don't know, maybe we're in a situation where we use a scalpel, we use some suture materials. You're thinking like bleach, alcohol and a flame.
Well, I love flame.
Boiling water.
Boiling water is really nice too. Yeah, we'll talk about all those physical methods and sterilization and disinfection tomorrow. But in an austere environment, I don't know, like I've never worked on the clinical side, but I was with an operational lab unit called the first AML, the first Area Medical Laboratory, which is a portable lab. We bring with us like our own incubators and PCR equipment and all that stuff, right? And we have to maintain, pretty clean environment wherever we are, which is difficult to do, so I can appreciate that. But we've adapted that lab so that we can maintain it in such a way that we need to under those conditions. We actually bring our own field configured autoclave with us as well, so we can use that autoclave to make media. You guys, you guys do what I don't know. Like I don't know what you guys do. I think, I mean, a lot of disposable stuff, right, a lot of one single use stuff probably, yeah, they're gonna turn more shit. Yes. Yes. Well, for one thing, somebody asked why prions are the most resistant. And for one thing, you can't kill them, so they retain their infective capability. Yeah. You would have to completely destroy them in order for them to lose their infectious process, which is what we talked about before, conversion, coming into contact with normal proteins. Yeah. Yes.
Are we gonna discuss as a group some of the questions, cause I noticed like a great change from your exam. So I was wondering if we
Oh,
could
yeah.
talk about that as a group, or we had to come to your office.
So we unfortunately, we are discouraged